Good afternoon. Welcome back to Virology. Well, we have completed the reproduction cycle in this course. This is the last discussion we're going to have, the infected cell. We're going to look at all the metabolic and biochemical events that happen in a cell to make it, to support virus replication, efficient virus replication. So far in this course, we've focused on viral gene expression, we've focused on genome replication, and the assembly of virus particles. And what we haven't talked about is that all of those depend on host systems, including metabolic systems, biosynthetic system, making the triphosphates and the lipids, signaling and trafficking systems. We've mentioned trafficking quite a bit on microtubules and actin, for example. But today I want to get into some mechanisms by which these events happen in an infected cell. We're going to talk about 4 different areas today in a cell. We're going to talk about signal transduction. I think you know what that is, but we will discuss how it's relevant to virus multiplication. We'll talk about gene expression. We've already talked about transcription, but we have not talked about the translation of mRNAs to protein. And the regulation of that step is quite subtle and nuanced. We're going to talk about that today. We're going to talk about metabolism. When a virus infects a cell, it's got to make a lot of things. The thermostat has to be turned up, and lots of new things are made. Lots of energy is made, lots of precursors. And we're going to talk about how that happens, and finally, how cellular organelles are remodeled, in particular the ER and the Golgi. In many virus-infected cells, they completely go away, and those lipids are used to make membranes for the replication and assembly of virus particles. So let's start with signal transduction. Signaling pathways are ways that cells use, now that we're talking about cells here, to sense their environment and respond to it. Every aspect of cell physiology and behavior depends on cell signal transduction pathways. And viruses can change signaling to promote their replication. So here on the left is a typical signaling pathway. This is the PI3K, AKT mTOR signaling pathway. And you can see it starts with plasma membrane receptors. And in general, when we have these pictures up, the green arrows means a plus stimulatory influence, and red is inhibition, as you can see in this key here. So binding of ligands to many cell surface receptors leads to activation of PI3 kinase and GTPases, and AKT is a central player here. There are inhibitors of the downstream events. So the downstream events are all stimulating proliferation, cell growth, and autophagy, in particular mRNA translation, as you can see here. And so there are inhibitors of that. These TSC1 and 2 are inhibitors, as you can see. So this inhibition is going to inhibit everything downstream. And this is what makes these pathways a little tricky. AKT inhibits these inhibitors. So when you inhibit the inhibitor, there's no more inhibition. So this red arrow would be gone, and everything downstream can happen. A little bit complicated, but just look at it. You can figure it out. Now, viruses can interfere at many of these steps. So these are all viral proteins here written out. The usual players, adenovirus, influenza, herpes viruses, etc. And some of them stimulate these pathways. So a virus infects a cell, it may not, the ligands for these stimulatory receptors may not be present, so the virus just goes downstream and pushes it so that you get more translation, for example. And some viruses inhibit parts of these, so these viruses will inhibit the TSC1, 2, but that's good because that's an inhibitor of downstream, so now the downstream events will happen. Okay, so that's just one aspect of signaling. This has to do with making proteins, making cells divide, and so forth. All right, now, in virus entry, signaling plays a role. So when viruses bind receptors, they typically initiate signaling pathways. So I have 3 here, 1, 2, 3, to illustrate. So on the left, we have adenovirus binding its receptor. That binding leads to uptake into a coated pit. And that binding to the second receptor here leads to initiation of a signaling pathway, these green arrows with all these proteins here, PI3 kinase and the Racaniello protein, RAC here. No, I didn't discover it. I didn't work on this, which stimulates, the green arrow means it stimulates the breakdown of the actin microfilaments, because when you have this mesh of actin under a vesicle, the vesicle can't pinch off. So that binding to the receptors stimulates the actins from coming apart, and then you can make an endosome and the virus can get in. So that's a beneficial thing for entry. Influenza virus also stimulates signaling pathways. The binding to receptors leads to activation, again, of similar pathways that allow the actin microfilaments to be disassembled so you can get an endosome here. Then there's also some clustering of receptor protein tyrosine kinases. Normally these are monomers. They cluster upon virus binding, and they help to activate these channels that pump protons into the endosome. So they're a normal part of endocytosis, where the endosome has to become acidified, and that signaling is involved in that. And then there's another signaling pathway. When the virus binds to its receptor sialic acid, it activates a calcium channel, which brings calcium into the cell and stimulates RNA synthesis. And finally, Ebola virus there on the right. You remember it's taken up by macropinocytosis, and it ends up in an endosome. This time the signaling has another kind of beneficial effect. So the binding binding to these cell surface proteins during macropinocytosis activates PI3K, AKT, and then RAC. RAC inhibits the endosome from fusing with lysosomes, which is a bad thing because the virus would be degraded. So you stop the trafficking at the endosome, and the virus then, you may not remember, but it binds an endosomal receptor and the nucleocapsid gets out, right? So this vesicle would be not permissive for fusion, so it doesn't happen because of the activation of this signaling pathway. 3 examples of how signaling are modified in virus-infected cells to stimulate entry. This PI3K mTOR relay is very important. Many viruses activate it to get more translation, hiked metabolism, as you can see here. AKT, also, its activation prevents apoptosis, programmed cell death, so that's good for the virus, right? So many viral proteins, as you can see here, they're these words. Here's the protein, hepatitis C, NS5A, rotavirus, NSP1, adenovirus, P110. These activate the AKT pathway, as does a cytomegalovirus pathway. So this leads to cell survival, not apoptosis, so that the cells live a long time so the virus can reproduce, and it activates translation and it hikes up metabolism. So these are beneficial for virus multiplication. And on the right, other examples of how viral proteins stimulate the AKT axis. Again, to get translation going, also to get cells to divide as well. And inhibit apoptosis. Apoptosis inhibition is really important for DNA viruses in particular because they tend to have longer reproduction cycles. And so they produce proteins that inhibit apoptosis. Although late in infection, apoptosis may occur, but that's fine because then the particles need to get out. And so the cell dying would not be an obstacle. Now here is an interesting twist where a virus RNA actually induces apoptosis. And this is a flavivirus. These are these envelope viruses where the glycoproteins are parallel to the envelope, and there's a plus-stranded RNA that encodes a long polyprotein, which is processed. The 5' end is an untranslated region or untranslated region, and so is the 3' end, okay? And that's shown schematically here on the left. So 5' UTR with an open reading frame. It's not to scale. It's much, much bigger than this. And then the 3' untranslated region. And a certain fraction of viral genomes, as the virus is replicating in cells, a certain fraction of viral genomes are degraded by RNases. We're going to talk more about these RNases in a moment, but one of them is called XRN1, and it chews up actually most of the genome. It chews up through the whole 5' UTR. It chews the whole open reading frame up, and then it hits this 3' UTR, and it can't go anymore. This structure blocks it from chewing. So you're left with this small flavivirus RNA. It's called sfRNA, because although the flaviviruses tend to do that. That sfRNA inactivates AKT. Let me remind you, AKT normally inactivates apoptosis. So if you inactivate AKT, you're going to turn on apoptosis. So this small flavi-RNA inactivates AKT and induces apoptosis. And this sfRNA is needed to make plaques, and it's needed for pathogenicity in mice. So here's the wild-type virus. It makes nice plaques on a monolayer of cells. If you make a deletion near the 3' end that prevents the production of sfRNA, you don't get any plaques. Now, this doesn't exist so that scientists can do plaque assays, right? It's somehow there to kill cells. That's what apoptosis is. So if you do apoptosis, you get a plaque. And so the bigger picture, what's the function of this? It's not clear to me. If you take this mutant virus and put it in mice, it has reduced pathogenicity. So this sfRNA is selected for some reason. I'm not clear, but this is an interesting example of how an RNA can inactivate the AKT, a protein. Now, gene expression is also inhibited. What do I mean by that? I mean transcription, essentially. The transcription of a DNA to form a pre-mRNA, followed by splicing and polyadenylation, nuclear export, translation, and eventually degradation. We'll talk about degradation separately in a moment. These red letters are all the ways that viruses inhibit these processes. Every step is inhibited by a viral protein. It's transcription and capping is inhibited, polyadenylation, splicing, RNA export, translation, as we'll see in a bit, and RNA degradation is inhibited or stimulating dependent on the situation. So you may ask, why is this happening, or what's the function? It reduces competition of cellular with viral RNAs. Now, there are some DNA viruses, of course, whose genomes go in the nucleus, and they need to be transcribed, right? So you can't shut off your own transcription. So those viruses have ways to get around this inhibition. Viruses interfere with splicing. And so here is a pre-mRNA in the nucleus with an exon, 2 exons and an intron. And all the splicing proteins are bound to it, right, to get ready to splice out the intron. A herpes simplex protein inhibits splicing. And then what else? What do we have? Then we have export of these spliced mRNAs. Remember, there are a number of splicing proteins that remain bound to the spliced mRNA, and those are signals for export through the nuclear pores. Viruses inhibit the export in a variety of ways. Influenza virus inhibits export. Poliovirus actually cleaves proteins associated with the nuclear pore called NUPs and inhibits export as well. So at every step, our pre-mRNA processing is inhibited as well. Again, to eliminate competition with cellular mRNAs. Now, mRNAs turn over in the cell. You know, when they made the mRNA vaccines for COVID, They put a signal in that would make sure it didn't last forever. And the people who say it lasts forever don't know this stuff. They need to take this course or something, because mRNA, depending on signals in the 3' end, has a very short, an intermediate, or a long half-life. The half-life is carried out by nucleases that are present in the cell. So here is a typical mRNA, poly A tail, 5' cap, open reading frame. There are a set of enzymes that remove the poly A tail. CCR4 not PAN. PAN is the only one I recognize. Poly A nuclease. That makes sense. I don't know what the others stand for, and it doesn't matter. It chews away the 3' poly A. And that allows both 5 to 3' and 3 to 5' decay. So there are specific enzymes for these processes. And before you can chew up from the 5' end, you have to take the cap off. So there are a series of proteins that do that, including decapping proteins. So there the cap is chopped off. And now XRN1, which we've met, goes and chews from the 5' end. There are also nucleases that chew from the 3' end. The exosome is one. It's a nice hat there, right? It's very cute. And it's chewing from the 3' end. So this is the way that the cell gets rid of mRNAs that need to be gotten rid of, whether it's after a short period or a long period. Now, in virus-infected cells, many of these proteins are either removed or relocalized so that they can't work, so they don't degrade virus-infected cells. And in some cases, like in polio-infected cells, these 3 nucleases, PAN, DCP1A, and XRN1, are degraded. So PAN is the poly A nuclease, and DCP is the decapping protein. And viral proteins can also initiate mRNA degradation. So here's an mRNA being translated. There's our ribosome. And remember, I don't think I taught you this, but we believe that mRNAs need to circularize in order to be efficiently translated. There's some interaction between the poly A binding protein, which binds the poly A, and the cap binding protein at the 5' end. The idea is that the ribosomes initiate translation at the 5' end, and then when they reach the 3' end, they can start over and just keep going around. Very efficient translation happens. You can translate without being a circle, but it's not efficient. So there's a circle being translated, and viral proteins can initiate degradation. So here we have an NSP1 protein of SARS-CoV-1 and a SOX protein of HHV8. They are chewing up the RNA. And then XRN1 can take over and degrade the rest of it. Herpes virus can also initiate degradation here as well. The vaccinia virus encodes proteins that remove the 5' cap. D9 and 10 remove the cap, and then XRN1 can chew away as well. So this is a way that viral proteins take the cellular mRNA degradation pathway and turn it towards cellular messages. Now, again, they don't do it against their own messages. That wouldn't make any sense, right? So there are specific sequences that prevent viral messages from being degraded for every virus. All right, let's have a quiz question here. Which of the following is a consequence of viral proteins modifying signal transduction pathways to promote replication? So 1A, polio inhibition of transcription by RNA Pol II. B, herpes virus protein blocking pre-mRNA splicing. C, disruption of actin filaments to allow endocytosis. D, initiation of mRNA degradation by viral proteins. So which one involves signal transduction? All right, let's have a look at the results. So most of you got disruption of actin filaments to allow endocytosis. That's correct. So that's a signaling pathway. Right? When the virus binds its receptor, it signals to disrupt the actin filaments. The inhibition of transcription, the protein blocking splicing, the degradation, those are not initiated by signaling pathways. It's just that one. I'll show you. let's go back to this. So here, in two cases, we have disruption of actin microfilaments by signaling pathways. So these signaling pathways involve binding to receptors and then signals going, these green arrows, to various protein complexes. Whereas these inhibitions of gene expression, they're just the production of viral proteins which then inhibit the cellular process. Now, you remember in the Baltimore scheme, this may be the last time we see the Baltimore scheme. Really sad moment. The mRNA is at the middle. We haven't talked much about mRNA. All these viruses need to make mRNA that can be translated by the host cell, and this process is highly manipulated by virus infection. So an mRNA looks like this diagram at the top. So the green is the mRNA. There's the cap, and there's the AUG where translation begins. And the way ribosomes are brought to this is through this big protein complex. So there's a protein called EIF4E, which binds the cap, and then 4E binds 4G, and then 4G binds 3, and then 3 binds the 40S subunit. So that big assembly of proteins binds to the 5' cap. And then it starts to move down the RNA until it reaches an AUG. ATP is hydrolyzed. Many of these proteins are released, and in comes, well, it's already there. Here sits the initiator tRNA, right? It's going to recognize that AUG. It's a met tRNA with a couple of proteins and GTP attached to it. So that starts translation. And what happens next is that the 60S subunit joins. So once the initiator is there, the 60S subunit joins, and then it can start reading the codons as it moves down the RNA and add amino acids here. But many proteins participate in this initiation process. You can see many of them are lost when the 60S subunit is added. Now, many viruses inhibit translation of cellular proteins specifically so that they can have the entire translational apparatus for themselves and have very high efficiency translation of viral proteins. So this is an experiment from my lab many years ago where we infect cells with poliovirus and then we measure protein synthesis at different times after infection. And if you just look at uninfected cells, there's a relatively constant rate of protein synthesis. But in poliovirus infected cells, by 2 hours, it's crashed. It's almost down to nothing. And then it goes up, and that is viral protein synthesis. So the cellular protein synthesis is turned off. It's replaced by viral protein synthesis, and then the cells die, and they can't translate anymore. On the right is a protein gel that we use to look at this, and these are time points after poliovirus infection in hours. And we've labeled the cells for a brief period with S35 methionine to label proteins. And you can see initially there's a lot of cellular protein synthesis, lots of proteins made in cells. But slowly those go away. You see by 3 hours, most of them have been turned off and they're replaced by viral proteins, which are these ones that are labeled here. So this is very typical of many viruses. They inhibit specifically cellular translation. You can see the viral proteins continue to be made, right? So it's a specific inhibition of cellular translation. How does this work? There are a number of mechanisms, as you might expect. Now remember, I'll remind you that in order to translate, the cap binding protein has to bind the cap and recruit all these other proteins in, which eventually bring in the 40S subunit. We call this 5' end dependent initiation because that cap is at the 5' end. Now, some viruses encode proteases that cleave EIF4G. Poliovirus, foot and mouth disease virus, and others will cleave EIF4G. Now, here's the cleavage point here, this little jig-jag line. With 4G cleaved, you can no longer recruit the 40S subunit to the RNA because even though 4E binds, it's not attached to EIF4G, right? It's cut off. So that's how shutoff occurs there. There is also a dephosphorylation of EIF4E. So for 4E to bind efficiently, it should be phosphorylated, and some viruses induce dephosphorylation of EIF4E. And then finally, other viruses induce dephosphorylation of a cell protein. So there's a cell protein called 4E-BP1 that normally binds 4E and inhibits it. So it inhibits it from binding the cap. 4E-BP1 is phosphorylated in the place where it would bind EIF4E. All right, so if it's dephosphorylated, then EIF4E will bind 4E-BP1, and then it can't bind EIF4G. So these are ways that viruses induce shutoff of cell protein synthesis. Now, there's another twist for many viruses is that in that the actual mechanism of initiation can vary. So here we have the canonical initiation, which I've just showed you, which happens for cell mRNAs and many viral mRNAs as well, 5' end dependent, right, where the 4E binds the cap, and then 4E recruits all these other proteins, eventually the 4DS subunit. Many viruses, including poliovirus, forget about the cap. They use what's called internal initiation. So the EIF4G actually binds directly to the 5' untranslated region, this highly structured region, so it can recruit the 4DS subunit. Now, you can see that this would happen even if 4G is cleaved, as it happens to be cleaved in cells infected with these viruses. So you cannot bind the cap. And in fact, the viral RNA, poliovirus RNA, doesn't have a cap, but rather EIF4G, cleaved or not, can bind this highly structured region. And then hepatitis C virus, a flavivirus, is even more unusual in that there's no cap, there's an internal ribosome entry site, and that will bind EIF3 in the 4DS subunit without anything else. So these sequences, which I've depicted as squiggly RNAs, they can bind the initiation proteins directly. So they're called internal ribosome entry sites, or irises. And they allow poliovirus, for example, to be translated without a cap. There's VPG at the 5' end, if you remember. So here's how it works. Here's the poliovirus genome. This is the sequence here at the top. This is, I actually did this sequence in 1980. It took me a year to do this genome sequence, 7,440 bases. And I had to type it out by hand. And then to make this figure, I cut strips of the typed letters out and glued them on a big board and took a picture of it. That's why this is so fuzzy. Because nowadays nowadays it would be done in a half hour and the computer would spit out this sequence. We couldn't do it. But here's the methionine initiation codon. It's about 742 base, and this is all 5' untranslated region. And it turns out that this is the internal ribosome entry site. It's highly structured and allows ribosomes to bind in the absence of a cap. And this translation occurs of the whole open reading frame, and it leads to the production of what we call a polyprotein, 3 proteins joined together, and then these are cleaved by proteases to make the final proteins, including the capsid proteins, which you've seen before, and the 3D Pol, the RNA polymerase. So these sequences are present in many viruses, and they're even present in cellular mRNAs, but they're pretty rare, and we put them into categories because these sequences, here is the poliovirus iris that I've talked a little bit about. You can see it's highly structured, and it's not like the iris of any of these other viruses. The sequences are totally different, and the structures are different. So we have 6 categories of viruses. One that's notable here is the type 2, where the EIF4G footprint has been mapped. In other words, where EIF4G binds to the iris, which I showed you here, that's been mapped specifically to this particular part of the structure here. So the idea is that the ribosome would bind there and then scan to reach this AUG. Some of these others are interesting. This one is a type 6, which is found in the genome of hepatitis C virus. This is the type 3 iris here, where the AUG is actually part of the iris. This one is an insect iris where the initiation codon is not methionine, but a proline. Different kinds of viruses. In the end, they all allow the ribosome to bind internally. So that's one way that viruses can direct the translational apparatus to their mRNAs. They inhibit the cell, mRNA translation, but they have some kind of mRNA that has unique requirements that can continue to be translated. But the cell doesn't just sit around and allow itself to be manipulated. The cell also fights back. And here's one way it does so. So initiation of translation, that step where the ribosome is binding to the 5' cap, this is highly regulated. And this is shown in this slide. So there's that initiation complex, right? We have our MET tRNA, we have the 40S subunit, and what is going to happen next is that the 60S subunit is going to bind, and then this GTP on the initiation, on this MET tRNA is hydrolyzed to GDP, as you can see here, and this protein GTP-EIF2 is released. This needs to be made back to GTP, okay, because you cannot have a new ternary complex come on the next mRNA unless you have GTP-EIF2 bound to it. And EIF2 is limiting in the cell, so this GDP has to be recycled for GTP. And that's done by a protein called EIF2B. EIF2B grabs these two, EIF2 and GDP, and exchanges the GDP for GTP. So it's a guanine exchange factor, or guanine exchange protein. So the EIF2 stays in there, the GDP goes off, the GTP is substituted, and then the whole thing is released so it can join up with the new MET tRNA and do another round of initiation. Okay, so this is how you get multiple rounds of initiation. This is a point of intense regulation in the cell. In fact, there are a number of protein kinases, EIF2 alpha kinases. So EIF2 alpha is a subunit of EIF2. These proteins, in response to various conditions in the cell, will phosphorylate EIF2, as shown here. And now, when it binds EIF2B, it's stuck. It can't be exchanged for GTP. And so eventually, all of the EIF2B is sopped up and translation stops. And the conditions that lead to this include ER stress. So when a lot of misfolded proteins are made in the ER, there's a sensor that picks that up. It's called PERK. And it phosphorylates EIF2 alpha. If you deprive cells of amino acids, there's another kinase in the cell, GCN2, that phosphorylates EIF2. And then, in the presence of double-stranded RNA, another kinase, PKR, leads to phosphorylation of EIF2 alpha. These are all conditions where you have to shut off translation. Misfolded proteins, if something's wrong, you have to stop translation until you figure out what's wrong, and that's the whole idea. If there's no amino acids, you got to stop translation. But what's double-stranded RNA? That comes from virus infection. Cells don't typically have double-stranded RNA, but when viruses infect them, it accumulates, it's sensed by PKR, and the cell will shut down translation so that virus reproduction can't occur. So every virus on the planet has to get around this, otherwise it doesn't exist. So I'll tell you some ways this happens. This is how, first of all, this is how PKR is activated by double-stranded RNA. PKR is produced in a cell as an inactive molecule. It has a kinase domain, KD, and it has two double-stranded RNA binding motifs. So these are produced in an inactive form. There's actually a protein in the cell called PACT, which will activate them, so these have cellular functions. But if you make double-stranded RNA, as in a virus-infected cell, this will bind two molecules of PKR. They will phosphorylate each other, and now they are active. But the key is the RNA has to bind two molecules of PKR, not one, because they have to phosphorylate each other. So this is how double-stranded RNA is sensed. PKR is activated, and then that leads to the shutdown of protein synthesis. Okay? Now, PKR is a very interesting protein. It's actually induced and activated by virus infection. I've told you how it's activated by double-stranded RNA. It's induced by interferon, which we're going to talk about in a couple of sessions. It's one of the interferon-induced proteins. So interferon is an antiviral defense. So this is part of the antiviral defense. And as I've shown you, it leads to phosphorylation of EIF2 alpha, which shuts down host translation. It will also lead to apoptosis. Phosphorylation of EIF2 alpha leads to apoptosis, which is programmed cell death. So basically, the cell is killing itself to prevent virus multiplication. But viruses have evolved many mechanisms to inactivate this pathway. Yes?
Is the double-stranded RNA virus you talked about previously, is that why it stays in the capsid?
That's one mechanism for evading PKR, to stay in the capsid. Yes, not all viruses do that, but they have other mechanisms for evading PKR. Yeah, which I will show you. So here is one by adenovirus. So adenovirus is a DNA virus. So where's double-stranded RNA coming from? Well, both strands are transcribed. And sometimes you're going to get complementary RNAs made that will hybridize and make a double-stranded RNA. And so what happens is you get double-stranded RNA in adenovirus-infected cells. It binds two molecules of PKR. They phosphorylate and activate each other, phosphorylates EIF2 alpha, protein synthesis inhibited. So early in adeno infection, you can see protein synthesis going down. But then the virus makes a small RNA called VARNA1. This binds one molecule of PKR. So there's VARNA1. It binds one molecule of PKR, which is in its inactive state, and it can't be activated because it needs to be next to another PKR in order to be activated. So VARNA1 restores protein synthesis because EIF2 alpha is no longer phosphorylated. That's one example of how a viral protein counters the effect of PKR. It prevents its activation. So here are some proteins of other viruses that counter the inactivation of EIF2 by PKR. So there are some proteins. The one I've just told you, adenovirus VA1, is an antagonist of RNA. It binds the PKR, so it basically only binds one, prevents phosphorylation. Epstein-Barr virus also has a similar protein. There are proteins that antagonize the protein itself. There are also double-stranded RNA binding proteins. So viruses that make double-stranded RNA, they make proteins that code it so that VARNA cannot bind to it, and it won't be activated. There are also inhibitors of the kinases. Here's PERK as an example. Viruses make proteins that inhibit the kinase, and then there are also inhibitors of EIF2. Vaccinia virus in particular makes a pseudo-substrate. It's a protein that looks like EIF2 alpha, and PKR will bind it, but it doesn't get phosphorylated and it doesn't have any activity, so it binds up all the PKR. So if you do not have an antagonist, you're not a virus because you've been eliminated a long time ago. All right, next question. PKR is an interferon-induced enzyme that is activated by something leading to phosphorylation of something else and inhibition of translation. So GDP, EIF2 alpha, double-stranded RNA, EIF2 alpha, double-stranded RNA, EIF2B, single-stranded RNA, EIF2 alpha, or none of the above. Fill in the blanks. Okay, let's see how we did. Right, most of you got B. So it's activated, PKR is activated by double-stranded RNA, which then phosphorylates EIF2 alpha. Metabolism. Do you know what that is? Anybody remember what that is? Well, if you asked me in college, I wouldn't either. The Krebs cycle, the TCA cycle, which I learned in senior biology, and I didn't understand any of it. And only when I started studying viruses did I finally. So viruses, to make a lot of viruses, thousands of virus particles per cell, you need components, right? And so the cell has to step up. You need nucleotides, amino acids, fatty acids, and energy. You need 4 ATP to make one peptide bond, methionine to alanine, 4 ATP, and this is cranking along thousands of amino acids per second. Oh my gosh, you need a lot of energy. So virus infection turns up cell metabolism, something actually we didn't appreciate for many years. It's only recently recognized that this is the case. And this is the TCA cycle, which we're going to come back to talk about what viruses do. So here's an illustration of viruses, what they do. So here we have cells infected with adenovirus. We have mock-infected cells, adeno wild type, and a deletion of E4 that prevents replication. It's a gene, an early gene. And these are little six well plates, and they have a red dye in them, which is phenol red that turns yellow when the medium gets acidic. And you use this as an indicator to make sure your cells aren't too acidic. So look what's happening in the wild-type virus-infected cells. By 48, 72 hours, the medium is turning acidic. The pH is dropping because these cells are producing lactic acid, which is the end product, I believe, of glycolysis. So adenovirus is turning up the glycolytic pathway to get more energy. And the E4 mutant, which doesn't replicate, doesn't do that at all. And on the right is just a quantification of glucose consumption. And you can see red is the wild type and blue is the mutant. So there's an increase in the consumption of glucose. And many viruses have a sweet tooth. Rhinoviruses are another one. So what is this sweet tooth? Glucose metabolism. So you eat carbs, and glucose is a major breakdown product of those carbs. And glucose circulates, and it's taken up into cells by the GLUT transporter. There's the plasma membrane. This is the cell now. So now we have glucose in the cell, and it undergoes a series of metabolic processes. Eventually, it's converted to pyruvate, yielding 2 ATP and 2 NADH, and there are more intermediates that allow you to get even more ATP synthesis. And then at the end, you have lactic acid. There you go, lactate dehydrogenase. And that is acidic, of course, and makes the medium blue. So in addition to the ATP and the NADH that are used for energy, you can also take glucose 6-phosphate and move it off to the pentose phosphate pathway. And this can be used to make nucleotides, right? You need a ribose in every base, so that's where that ribose is coming from. And then acetyl-CoA is a product of pyruvate dehydrogenase metabolizing pyruvate, and that is a precursor for fatty acid synthesis. So viruses crank up this pathway because they get a lot of energy, they get precursors to nucleotides, and they get precursors to fatty acid, right? And different viruses have different effects on glycolysis. Here again is an abbreviated form of our glycolytic pathway on the left. And we have 2 herpes viruses here, human cytomegalovirus and herpes simplex 1. HCMV stimulates the uptake of glucose. The green arrow and goes all the way to, pushes it all the way to acetyl-CoA, the precursor for fatty acid synthesis. But herpes simplex virus doesn't really increase glucose uptake. It allows for the steady state to occur, and it inhibits the processing of G6P to pyruvate and instead shunts off G6P to the pathway for making nucleotides. So CMV likes fatty acids, HSV nucleotides. And the hypothesis is that because HSV has a shorter reproduction cycle, it needs a lot of nucleotide precursors to synthesize viral DNAs. Fatty acid synthesis is also tweaked, as I implied. Now, here we have pyruvate, right, which is the product of glucose metabolism. Has to be converted to acetyl-CoA. But acetyl-CoA can't cross the mitochondrial membrane. That conversion occurs in the mitochondria, pyruvate to acetyl-CoA, and it can't cross the membrane. So it is converted to citrate. Citrate can leave the mitochondria, and it's converted back to acetyl-CoA. That's the way metabolism evolved. And some viruses can interfere with this pathway. So most of the citrate in HCMV infected cells are shunted to fatty acid synthesis here. So remember, HCMV needs a lot of fatty acids. And if you inhibit fatty acid synthase, you get a reduction of HCMV particles. And another member of this shuttle is also increased in cells infected with VSV. So this is the way you make fatty acids, and this is just a brief two examples of how viruses interfere with it. Here's the citric acid, or the TCA cycle, which of course is a major hub of carbon metabolism. It gives you precursors for many compounds, and viruses impact this cycle in a major way. So the input, of course, is pyruvate, which then is converted to citrate and then converted to a series of other compounds, eventually leading back to citrate. And along the way, generates a lot of energy. Now, viruses tap into this pathway at multiple steps. So HCMV takes the citrate out of the cycle to make fatty acids, right? So there's your citrate there. HCMV steals that. But it puts back glutamine down here, otherwise the cycle would stop. If it kept pulling out citrate, there'd be no TCA cycle, no energy, and so forth. So HCMV, vaccinia virus, simplex virus, they all do this. HSV does something separate. They pull glutamine in to prevent halting of the TCA cycle. And then HSV pulls off oxaloacetate, changes it to aspartate, which are eventually converted to pyrimidines and purines, which are made for nucleic acids, right? And again, consistent with this idea that herpes simplex needs a lot of nucleotides, this short replication cycle. Now, a little bit more on lipids. I think it'll come into play later. The primary store of lipids are triacylglycerols, right? So this is a triacylglycerol with 3 lipids. And this is how lipids are stored. And it's also an energy source. So you can oxidize lipids and get lots of energy. So you can do that with glucose, or you can do that with lipid. And in the blood, lipids circulate as lipoprotein complexes. When they're needed in cells, the lipoprotein lipase makes free fatty acids from it, which are imported into the cell, converted to acyl-CoA, not acetyl-CoA, acyl-CoA, and then triacylglycerols, like these, and then stored in lipid droplets. So in the cell, these lipids are stored as these droplets. So the lipid is yellow and the protein is blue. So viruses modulate lipid biosynthesis. They have to modulate the synthesis of lipids as well as its oxidation. So have a look at that in the next couple of slides. Now, cytomegalovirus infection needs very long chain fatty acids. What do I mean by that? So these are the different the carbon length of the fatty acids in a lipid, right? So these are of a certain length, and they can get very, very long, and cytomegalovirus likes the long ones. And so what HCMV infection does is increase the passage from glucose to acetyl-CoA, which I already told you, right? And this long-chain fatty acid is not only needed to make viral proteins, but it's required for infectivity. And the way it works is shown here. It's a complicated pathway. It involves turning up activators of transcriptions of genes for lipid synthesis. So let's start all the way at the end. Here are some genes that are needed for fatty acid and sterol synthesis. They're transcribed. The HCMV has to turn them on. There is a transcription factor called SREBP1M that turns on the transcription of these genes. So HCMV has to turn that on. So we'll go back to the beginning again. SREBP1 is in the ER. It's held in the ER. There's PERK, just to remind you of your old friend. So here's the ER membrane. And SREPB1, this is it right here, is held in the membrane. When you have infection with HCMV, it stimulates, it makes glycoproteins, of course, it activates PERK, and it stimulates the release of SREBP1 from the ER into vesicles. It proceeds through the Golgi where the active part gets cut off. So you see these two transmembrane portions serve to anchor it through the Golgi, and then once you've reached the far part of the Golgi, it can be cleaved, and now you have the SREBP1, which goes in the nucleus and turns on fatty acid synthesis. And then normally, a protein called lipin-1 inhibits the import of this transcription factor into the nucleus. That inhibition is inhibited by mTORC. So HCMV turns up mTORC. These are signaling pathways which inhibit lipin and let SREBP1 get into the nucleus. So the whole point of this is that HCMV needs more lipids, so it needs to turn on their synthesis via this transcription factor. Pretty cool, I think. Hepatitis C virus, which we'll come back to in the next weeks, is a very important human pathogen, increases accumulation of fatty acids in infected cells. So here's an experiment where we have human hepatocytes in culture. We have them mock infected, and they are stained with DAPI to show the nucleus, and with a dye that stains lipids green. So you can see there's lipids in uninfected cells. When you infect hepatocytes with HCV, you get much more lipids being made, as you can see, because they're needed for the replication and assembly of hepatitis C virus. So there has to be a balance between the synthesis of lipids, lipogenesis, and the oxidation. So what HCV does is to turn up lipogenesis by inducing this transcription factor, FOXO1, very much like the HCMV induction of transcription factor. More glucose will lead to the synthesis of more lipids, and then it churns down the synthesis of another transcription factor that is needed for synthesis of the genes involved in fatty acid oxidation. So here's FOXA2, transcribing genes involved in fatty acid oxidation, so HCV inhibits it. And if you experimentally overproduce FOXA2 in HCV-infected cells, you inhibit HCV replication. Now, the synthesis is shown on the left. FOXO1 is the transcription factor that is needed to make the genes involved in lipogenesis, including inducing SREBP1, our friend from the previous slide, fatty acid synthase, leading to the synthesis of lipids. And so HCV infection induces FOXO1. It also prevents FOXO1 from exiting the nucleus. It makes it stay there so that it can transcribe these genes. And if you knock down FOXO1 expression, say by siRNA, you inhibit hep C replication. So just an example of the importance of lipids and the balance between oxidation and lipogenesis for viral replication. Well, how might virus infection lead to increased levels of ATP? Stimulation of glucose uptake, increased glycolysis, increased oxidation of fatty acids, increased utilization of glutamine, all of the above. All right, let's see. Almost. Yeah, most of you got it. All of the above. Right. Virus leads to increased levels by stimulating glucose uptake, increasing glycolysis, the oxidation of fatty acids, gives you energy, and the pulling in of glutamine into the TCA cycle, keeps it going, and that makes energy. It's absolutely right. Good. All right, finally, last little bit of discussion, remodeling of cellular organelles. What does this mean? Cellular organelles include Golgi and endoplasmic reticulum. And in many virus-infected cells, after a few hours, those are gone, and they appear, the lipids in them appear as little vesicles that are used for virus RNA synthesis and eventually assembly of virus particles. And here's the example in poliovirus-infected cells. So again, we're staining cells for fatty acids, and here's a mock-infected cell, nucleus, fatty acids in the cytosol. Poliovirus infected, many more fatty acids in the cytosol, as you can see. And so what happens is there's a viral protein that grabs triglycerides from lipid droplets. So remember, these lipid droplets are the storage components for triglycerides. These proteins pull them to replication compartments and take out the triglycerides and make bigger compartments. And that's illustrated on the right here. So you have a poliovirus-infected cell, and these little vesicles begin to form. And again, those are made from lipid droplets, but also from ER and Golgi. They form into double membrane particles. And on their surface, this green is the RNA replication. So the RNA replicates on the surface of those membranous compartments. And eventually they become double membraned. They may actually capture some virus particles, as I suggested when we talked about assembly. And so they could release particles in non-lytic events. But the point of this is that the RNA synthesis and the assembly of particles takes place on these membranous complexes that are co-opted from cellular compartments. It doesn't just happen for poliovirus. Happens for many other RNA viruses, probably all RNA viruses as well. So here's hepatitis C virus. It induces these membranous compartments that are different from that of poliovirus, and that they are attached to the endoplasmic reticulum. And so eventually in virus-infected cells, you have this big network of vesicles attached to the ER. And these vesicles are open, and they contain lots of viral proteins, including the RNA polymerase, and the RNA is synthesized in here. And eventually, lipid droplets are pulled in, and these contribute to the assembly of a virus particle. So here is a virus budding into this replication compartment. It's going to eventually bud in here. It's got RNA in it, as you can see here. It's got structural proteins, and it has the spike proteins embedded in the membrane. So the virus induces these structures just to assemble virus particles. And so you can see lipid droplets are very important for the reproduction of hepatitis C virus. Same with coronaviruses. Coronaviruses induce double membrane vesicles, not attached to the ER like hep C, but double membrane vesicles on which RNA synthesis occurs. Both mRNA synthesis and genome replication occur on these double membrane vesicles. Here is a cryo-EM reconstruction of these double membrane vesicles. You can see there are many of them in clusters. And again, they're derived from cellular membranes and from cellular lipids. And it's on them that the genome replicates. But the virus assembles on the ergic, as I mentioned before. It does not assemble on these like HCV does. And finally, There's a very cool example from plant viruses. So this is a virus of a plant. So it's a virus that infects tomatoes. It makes them bushy and short. So it's tomato bushy stunt virus. The name has been changed lately because people screw it up all the time. Now, this virus replicates its genome in vesicles attached to the ER. You can see viral RNA synthesis occurring here. That's the viral polymerase. And what this virus does, it's very interesting. It takes this viral protein, P33, which lines this vesicle, and P33 drags in pyruvate kinase, which makes ATP that's needed for viral RNA synthesis. So there's PEP in the vesicle. There's this pyruvate kinase is dragged in by its interaction with P33, and then it leads to the production of ATP, which is the polymerase is using to make, actually it's for the helicases to unwind the double-stranded viral RNA. And here's an experiment that shows that we have P33 labeled with blue fluorescent protein. So this is a virus infected cell. Then we have cells labeled with green pyruvate kinase. So they're engineered to have a GFP pyruvate kinase. So this is the vesicle right here that I've illustrated here. So you can see the P33 is there. So is pyruvate kinase. And when you merge the two, you can see they're both in this vesicle. So that's the idea that P33 is dragging pyruvate kinase into this vesicle so that it can make energy. I think this is pretty cool that it does that. Anyway, lots of metabolic manipulations in virus-infected cells. Those are just a little sampling of them to give you an idea what goes on. Now, next time, we're going to start the second half of this course, which is all about disease. And we'll start with talking about infection basics.